All right, so today we're going to be looking at karyotypes and sex-linked traits. All right, so humans are going to have 23 pairs of chromosomes. So total, there's going to be 46 individual chromosomes. The first 22 pairs of chromosomes are autosomes, or the chromosomes that do not code for gender. So the first 23. Okay, so basically everything up until this point here is going to be autosomal, which means they code for everything except for gender. So they're going to code for things like hair color, height, personality characteristics, eye color, everything that codes for anything except for gender. Now, that 23rd chromosome is going to determine the gender of the offspring. So if you're female, you're going to have two X's, and then if you're male, you're going to have an X as well as a Y chromosome. So this is what doctors basically do when the parents want to figure out the gender of their child. They can do a karyotype analysis and they can figure out the gender, but they can also figure out if there's going to be any abnormalities in the chromosomes. So for example, Down syndrome, they're going to have a triple or extra chromosome, and they're able to kind of look at the chromosomes and determine if there's going to be any birth deformities or defects, as well as the gender of the child. All right, so when you're looking at the sex determination, you're going to have your male up top there. You're going to have your female here to the side. You do your crosses. XX is going to be female. XY is going to be male. So there's basically a 50-50 chance that you're going to either have a male or a female. So basically, what is a karyotype? A karyotype is going to be a chart, as we talked about before, of chromosome that's going to be paired in order by size, and then they're going to be arranged according to the length and location of their centromere. So you're basically just organizing. You're going to have 46 chromosomes going to be spread out in that nucleus, and we're organizing the chromosomes based on shape and the location of the centromere, and it's also going to have an effect or determine what this child is going to look like and what characteristics or physical characteristics this child is going to have. So what are they used for? They're basically used to determine the gender of an offspring, and they can also be used to pinpoint unusual chromosome numbers in cells. Okay, so this is what normal is going to look like, okay? So here, when we look at, this is a normal female. So if you look at chromosome number 23, there are going to be two X's. So that's how we know it's a female. And then here, if we look at the male, we're going to have an X as well as a Y for chromosome number 23. So we know it's a male. Everything looks basically around the same size. There's no chromosomes missing. There's nothing doubled. They're both the same size and shape. So this is how we know we're going to have a normal male or female. When we look at so remember that males have an X and Y chromosome at number 23, and females have both X's. Okay, so now when we look at something that's abnormal, all right? So here, if we look at chromosome number 21, there is an extra chromosome. Remember, they're only supposed to be pairs. So here there's a number 3. So we got trisomy. Tri means 3. 3 chromosomes. So we know that something is going to be wrong there. Okay, so non-disjunction. So basically, non-disjunction occurs when sister chromosomes fail to separate properly during meiosis. This results in too many or too few chromosomes in eggs and sperms. Okay, so this is basically what non-disjunction happens. So basically, remember in meiosis, the chromosomes need to double up. They need to join together at the centromere. During metaphase, anaphase, they pull apart. So during this process, something happens where these chromosomes don't separate properly, and then as a result, there's an extra chromosome in one cell, and then there's a chromosome missing in another cell. So sex-linked inheritance. These are traits that are controlled by genes located on the sex chromosomes. These are called sex-linked traits. So all the other Punnett squares that we've been doing, those dealt with autosomal chromosomes. So these are chromosomes numbered 1 through 22. Now we're looking at all the Punnett squares that deal with, or genetics that deal with the chromosomes located, or the traits, alleles, located on chromosome number 23, which is our sex-linked traits. So every time we're looking at sex-linked examples, it's always going to be written with the superscript of, XY, depending on if it's male or female, and then the trait is going to be a superscript right over that XY. So for example, if we're looking at, let's say hair color was an example of a sex-linked trait. So we have hair color of brown. So big B is brown, or little b is going to be blonde. If it was a female, we're going to have XX. A female with brown hair, homozygous, it's going to be big B. Okay? And then if it was a male, okay, it would be, let's say, male with blonde hair. It's going to be X little b. So the alleles, and we're going to talk about that a little bit later, but the alleles are always only located on the X chromosome. They will never be located on the Y chromosome. Okay, so female is going to be, if it's brown haired, X big B, and then for males, if it had blonde hair, it's going to be X little b, Y. All right, so here is our Punnett square for sex-linked trait. So females, we're going to have three different possibilities. We're going to have normal, which is, for example, if this trait was a sex-linked trait and it was a recessive sex-linked trait, that means that you need to have two recessive, or one or two recessive alleles in order for it to be expressed. So for females, because they have two X's, we need to have two recessive alleles in order for it to be expressed. And you can see that. And you can see that here, that we're looking at hemophilia. The only time hemophilia is expressed in females is if they have two of the recessive traits. Males, on the other hand, as long as they have that, because they only have one X chromosome, as long as they have that one recessive allele or dominant allele, depending on if it's a recessive or dominant condition, it's going to be expressed. Okay, so that's why males are more likely to get these sex-linked traits. Color blindness is an example of a sex-linked trait. Males are more likely to have this because of the fact that they only have one X chromosome. All right, so this is just an example of how you would set up your Punnett squares. Okay, so when you're doing your Punnett square, step 1, write down male or female. And then step 2, write down your alleles. Remember that only females are going to have two X's and the males are only going to have one X accompanied by a Y. But remember that only the X is going to have that allele attached to it.